Yeah, really the kind of first question, why we need kind of biocarrier? What is biocarrier about? Yeah, the biocarrier usually it's kind of, I don't find a good kind of definition in a, not in a paper, not in a Wikipedia, not somewhere, but we can say that it's kind of carrier when we deliver cells or bacteria kind of living materials. I mean, in a kind of micro or nano level. It's what I use as kind of definition for in my presentation. Why we need to use such carrier? Carrier, by the definition, is something which we need to deliver somewhere. It means that we can deliver it in a body or for tissue engineering or for detect something or for analysis or for assembly to create a new objects. Really, biocarrier, it could be or colloids, colloidal particles or colloidal system, or kind of films or layers on substrate, which can contain also the kind of cells or bacteria or kind of microorganisms materials. Yeah, really, if you have a look in kind of general biocarrier components, it will be kind of hydrogel, minerals, kind of biomolecules which we can functionalize it, or more important part, it is or cells or bacteria, viruses, or another kind of microorganisms. But major limitation, how this of all these materials we can combine together easily, but major limitation of them, how will be cells or bacteria as more sensitive part of whole system will be interact by the carrier by themselves, how it will be functionalized and how it will be stable in it. Yeah, in the case really we need to reorganize our kind of materials to design kind of successful biocarrier. In the case we can use the hydrogel as a matrix, as a kind of substrate for small flexible materials. We can fill them with minerals, with the biomolecules to add more kind of structural property or more function. And also we need to add the kind of this microorganisms, cells, bacteria, and so on. Really, I will start my talk about these materials, about the hydrogels, because hydrogel kind of usually more flexible materials. It's possible to use as a matrix. It's already widely used in drug delivery, in food, in wound healing, in contact lenses. But in my talk, I will be explain how we use the hydrogel as a biocarrier or as part of biocarrier on example of bone and tissue engineering. Why really bone and tissue engineering? Because it's quite interesting and kind of reasonable application, which knowledge from this area we can transfer later on to the bacteria or to create another substance. But here it is, for biomaterials, for tissue engineering, really we need more or less finalize all our goals. We need kind of materials which will load cells or have free cells, which we deliver inside the body, and cells should be stay alive in it and keep their functionality. In the case we need study the strong interaction between our carriers and between the cell, between our carriers and between such cells. And if we study, if we know how they cells proliferate, how they functionalize in such system, we can understand the limitation and the way of using such materials. But really, for creating biomaterials for tissue engineering, it's quite, yeah, in simple words, really there is kind of two tasks. We need to fill them as much minerals as possible. We need to add as much functional molecules as possible, and cells should stay alive and be functional in it. In the case when we create such materials, we can use them as a tissue implant or with a bone implant and carry them to the body. Yeah, really, if you go to the hydrogel to prepare kind of capsules, kind of materials, it's very easy. Really, all know, I mean, it's a kind of well-known recipe from molecular kitchen. If you take the liquor, you take sodium alginate, you take the calcium lactate, put another, mix the sodium alginate with the liquor and stabilize with the calcium lactate, you create a nice kind of caviar based on the liquor, which are very tasty and already very well-known materials. But Usually this idea works not only in the kind of molecular kitchen, not just for the kind of such capsules, gel preparation, but also in a kind of bioprinting. Bioprinting is simple and more well-known kind of biocarrier, really, which carry our cells inside the gels, and we can do the kind of positioning of our tip in the printing and create a different structure. In the case cell, after this proliferate and grows inside the gel matrix and fuse each other and formate the tissue. Really, such simple kind of effect of biocarrier, it's already exists, it's already applied a lot, and all can buy the kind of bioprinter. But the problem is that for bioprinting, usually use not just simple cell solutions, usually use the cell spheroids, because stability of the cell inside the gel matrix, it's quite complicated because it's not stable, it's not proliferate good. If you formate spheroid, it's work. If you add simple cells, you need add much more and other components, and you need solve much more other problems. Because really to cell proliferate in the gel matrix, yeah, it's not so easy because of the mechanical property, because they need supply of nutrients and many other problems. Yeah, in the case, we come in again, okay, if we like to use them kind of spheroids, so spheroids for cell organization, it's work. But if you'd like to have something complex, for example, load more free cells, not for tissue engine, but for a kind of another application, or we don't like use spheroids because also it's quite limited, we need to add maybe minerals that they support our cells to proliferate inside the gels. And really, here it is, we coming to the kind of mineral part. In our work, we mostly use a kind of, as example, calcium carbonate particles. And we need to study how these particles could be interact with the cells. But really, how we can create the kind of gels with the particles inside? There are a lot of kind of logical ways. We can create the particles and add them to the gels, or add gels to the particles. Or we can create the gels and mineralize them in it. And in all these materials look like they just kind of combination what we had first, particles or gels inside the particles, or particles on top of gels, have completely different way how they interact with the cells to formate final biomaterials which create the gels, particles, cells, and other function to deliver them. Yeah, and first part, it will be particles in cells. First of them need to understand what our particles about. Really, we mostly work with the calcium carbonate, and calcium carbonate exists in three modifications. Argonite, usually it is in kind of seashell, in kind of sea creatures shell, and so on. Calcite, which are most component of the chalk and look like cubic. But more interesting, it is vaterite. And vaterite really unstable, and in our laboratory we know how to synthesize them well. And why it's unstable? Because really I show here this photo of the water which I discovered, the vaterite in the ice area, because it's unstable in water. In the case we can keep them just in ice in a kind of conditions without free water. Yeah, but really the advantage of them that when it's in contact with water, it starts to be recrystallized to calcite. And in our materials, we can use this property. Really now the vaterite is possible to find in kind of ice areas. Sometimes it's in kind of squirt or in kind of sea plants and so on. But most of them are synthesized in the laboratory. Yeah, in our case, we just mix the two salts, calcium chloride, sodium carbonate, and in special condition, we can create the simple, cheap spherical particles. If we change conditions, for example, temperature or reaction time, we can synthesize different particles with different size, shapes, polymorphs, so on. And now we have in hand kind of spherical particles from 300 nanometers and now already from 100 nanometers to 12 microns. And since we know how to synthesize them, we can choose any size and can synthesize these particles. Also, sometimes it's useful to have elliptical particles or star-like particles, because all these particles really have completely different interaction with the cells, with biological materials through their geometry. Yeah, but since these particles are not stable, but they're porous. In this case, it's easy to load them with a drug. But when we, in the case, add the additional functionalization to our system. But really, when it's crystallized to the calcite, it's release a drug. In the case, we can utilize this instability of the waterite to the as drug release property. Okay, but really, to create kind of carrier from these particles, it's by themselves. It's very difficult because these particles it's quite small. It's up to 10 microns, and if you mix with the cells or with bacteria, they will be really not carry the cells or bacteria. They will be penetrate inside. Yeah, and such particles depend on size. It's easily penetrate inside if it's smaller than the kind of 1-2 microns. But really, if it's above than 2-3 microns, it will be mostly stuck on the wall. In the case, we can formate the shell around our biological objects. Also, as an option, we can modify our particles, for example, with magnetic nanoparticles. In the case, we can add the additional function to our biological objects.
The hand is raised. So let's see.
But where is it in the chat?
Here, chat. Here.
Yeah. Or yeah, for creating biocarrier, usually we'd like to have kind of hydrogel as a matrix, which will be kind of our structural components. Also, we can fill it with the minerals and biomolecules to provide additional kind of mechanical function, mechanical enhanced or another kind of function which you'd like. And also we add the kind of biological component as a cells or bacteria or viruses or something like this. Yeah. And in our work, really we'd like to focus on the hydrogels. I mean, we use kind of hydrogels as a structural component, and we'd like to focus about the creating biomaterials, biocarrier for the bone and tissue engineering based on the hydrogels and with additional functionalization with the minerals. And really, when we'd like to create kind of biomaterials for tissue engineering, we'd like to create kind of such hydrogels, which could be preloaded with the cells or could be cells-free, but will be suitable to the cells growth in it, which contain as much mineral as possible and as much functional molecules as possible, and as much kind of living cells as possible as well. How we can prepare kind of capsules from the kind of carrier or capsules from the hydrogel by themselves? It's quite easy. Really, it's possible by the kind of normal molecular kitchen technique. When you use a hydrogel, which are ionically cross-linked, you mix the kind of, for example, alginate with a substance which you need to encapsulate, for example, liquor. And after this, stabilize them or cross-link with the calcium ions. Calcium lactate, and as a result, you get the kind of caviar or kind of beads which contain our substance of interest. Really, unfortunately, it's yeah. This one idea, it is a kind of key idea of the bioprinting. Because if we mix our gels, for example, with cell spheroids, and start drop them in the crosslink solution, we can drop any possible, we can drop kind of possible tissue and possible structure. Yeah, and after this, really the spheroids can fuse and format the tissue. But unfortunately, it's not work so easy with the separate cells, because spheroid, it's kind of nice, stable system by themselves. But cell to growth, yeah, they need kind of source of media, they need kind of special mechanical surrounding, and they not easily grow in any hydrogels. In the case, but it's still you still don't see the slides or something. There is a problem with it. Can you answer? Because I saw. Well, come on again, not moving. Yeah. Yeah, what should we do? We can
switch the computer.
We can switch the computer. In the case here, wait kind of a few minutes. Yeah.
French.
Yeah.
Okay, so I will pause this.
Great. Okay. Yeah. Hello again. I will continue my talk about the hydrogel. In the case, it's from hydrogel, really it's quite easy to prepare the capsules. If you just by the pipetting mix your hydrogel with kind of substance and put to the crosslink solution, and they ferment kind of beads. But really, this one techniques quite commonly used for spheroids, fossil spheroids for bioprinting in bioprinter. Usually they premix with the gels, matrix, and start print to the crosslink solution. And if you put them close enough, they can fuse each other and ferment the real tissue. Because it's kind of simple example of the biocarrier. Because really we have here the kind of gel, the matrix, which we need delivered from the printer nose or from the kind of solution, and assemble to the tissue. Yeah, but really when we start work with the cells, this technique not work well, because really spheroid is kind of closed system and they stable by themselves and they can easily survive in a different matrix by themselves. But if we go to the single cells or single microorganism, I mean, it's not so easy because it's much more difficult conditions which will be around. For example, it should be enhanced mechanical properties. It should be source of the kind of growth media with nutrients which provide correct metabolism and so on. In the case, we can try to create kind of materials which are kind of carrier, which can create as much function as possible, as much cells as possible, as much minerals also as possible, because minerals it's also useful for the bones in our case. Yeah, and in since we use with the bone, we work with the bones, it's quite natural to use the calcium carbonate, because the bones consist a lot of calcium and they will be used for hydroxyapatite. Also, calcium carbonate is nice materials because it's very cheap and very easy to produce, and they're porous and can absorb any materials. How we can really mix hydrogel with the particles, how create composite materials. There are kind of a lot of natural way how to do it. We can first synthesize the particles and add them to the hydrogel, or ferment hydrogel on top, or do the mineralization inside the hydrogel. Really, in our work, we try almost all possible configurations of this, and really depends how we can, which subcomponent we add first and which we add second. It will be influenced to the final material, final composites, which will be influenced to the material cell interaction. Yeah. First of them, we will try to talk about more simple stuff. It's particles and cells. If you have a look on the particles, it is a calcium carbonate exists in three modifications. It is aragonite, calcite, and vaterite. And aragonite usually it's and calcite not so interesting because they are not porous minerals, but it's quite easy found in nature. But the vaterite, it's possible to ferment the porous crystalline, but it is very difficult to find in nature. In the case I show here the photo of the German scientist, German geologist, which found them in alpine area. Why it's difficult to find in nature? Because in contact with the water, it starts to be produced the calcium ions, and which are assembled to the most stable phase of the calcite. In the case now, every time when we found the waterite in nature, it's usually big event in kind of, it's in Greenland, in ice part, or somewhere in a kind of sea organisms, or in kind of plants, and in very small amount. But every time when we found them, it means that a lot of very special conditions was around. Yeah, how we produce them and why we like to use them a lot, because it's very cheap and very simple. You can just mix the calcium chloride and sodium carbonate, and two simple salts which you can find in pharmacy or in normal supermarket sometimes. And all rest really just depends on conditions. If we change, for example, temperature and time, we can produce very completely different sizes and shapes. If you start to work with concentrations, with additives and so on, we can have much more variations. Yeah, and really now in our hand, in our laboratory, we developed many techniques how to produce such minerals. It could be porous spherical particles from kind of 100 nanometers really. It's not updated yet in the slide, till 12 microns and more. Or it could be elliptical particles in size from around 3 microns, or kind of star-like shapes. Also many other shapes also possible, but this one very reproducible. And depends on particle size and shape, it will be completely another interaction with the biological objects like the cells or bacteria. Yeah, but science particles as porous, really it's very easy to encapsulate something in it, because they just absorb more or less any small or big molecules easily. But since they are not stable, really the release process, it's a matter of stability and crystallization process of the particles. It means that if we load, for example, such particles with a model drug like rhodamine, for example, and we incubate them 5 days in the water, if it's small particles, they will be released in 5 days. If it's big particles, they will be released in 1 day, because big particles crystallize quicker and transfer quicker to the calcite form. This idea really very important for the following application to create biocarriers because of 2 points. One of them, we can use this calcium ions to stabilize the gel, to crosslink the gel. Second one, we can use our calcium carbonate materials to increase the functionality, because we can easily load something, and if we know condition around, we know when it will be released. In the case, we can load the enzymes or antibiotics or something like this and increase the functionality of the biocarrier. Yeah, but if you go to the particle-cell interaction, what we understand that the particles really easily penetrate inside the cells. In the case, it's difficult to say this particle is a carrier by themselves. It's more likely opposite, vice versa, that the cells is carrier particles. But if particles small enough, they penetrate, yes, and in high efficient amount, in the case, it's easy to use them as drug delivery. But if the particles will be big enough, like the 3-4 microns, they will be mostly stuck in cell membrane. In the case, also we can functionalize such particles with magnetic nanoparticles, and they can provide the movement such function. Yeah, but really also we can absorb create a hydrogel particles as a template of this. In the case, if we, for example, take the calcium carbonate, we load them with alkaline phosphatase enzyme, which used in bone reconstruction process, and we put the hydrogel on top and crosslink with the calcium ions and dissolve the core, we create a hydrogel which contains our enzymes, which we can add to another hydrogel or understand how the cells grow in it. Yeah, and if we have a look, what will be better for cells if we take all our possible particles, like the unloaded, loaded, hydrogels, not hydrogels, with alkaline phosphatase. Yeah, we understand that, okay, for bone reconstruction, we have just one goal. It doesn't matter more or less how the cells grow in it. It's more important how many hydroxyapatites is produced by the cells, how quick our bone will be reconstructed. And what we realize here, that the particles which are contained, for example, calcium carbonate and alkaline phosphatase together show you 2000 times better hydroxyapatite production respect to the control. And for this one is two reasons. One of them, it is a calcium, provides the calcium ions. Alkaline phosphatase also supports the use as a kind of functional molecules which accelerate the hydroxyapatite production. It means that such particles together with the kind of cells should provide correct function, and we don't need to care not only structural function, but also for absorption. Yeah, so like and so on, because in 3D it's very difficult to work. In the case we go to the 2D model of this, and really we try to understand how the particles work if we how the cells grows if we just do the gels, we do the mineralization, and we to condense gel to enhance the mechanical property, we heat them a bit and condense. Yeah, and really what we realize that in such system it is we have such a lot of parameters because we do the mineralization inside, and it's our technical parameters which we just control by our reaction. We just add the different percentage of the calcium and magnesium ions which could provide the particles. But as a result, all of them influence to the Young modulus of the particles, Young modulus of the hydrogel, Young modulus of the composite totally, size of the particles, mass of the particles. And really all of them are strongly influenced to the cells parameters, how the cell adhesion to the such system. In the case we also try to measure all these parameters like the cell area, cell density, cell viability, hydroxyapatite production, alkaline phosphatase activity, and so on. And what is the problem as a result? That we generate such amount of the parameters, and it's just difficult to understand and difficult to plot what's what depends on what, because technically we do the just 5 concentration, we just mix the calcium and magnesium in a few different concentration, but as a result influence as snowfall in kind of progression to the 34 really different parameters. Yeah, and here it is, we try to plot, understand what is better, what better, and by eye we can see that in calcium it is cells usually absorb better, but the hydroxyapatite sometimes better, and kind of it's a bit worse, and it's not so clear the dependence. And the same with the mechanical property, we do a lot of IFM study, atomic force microscopy study. We do the kind of with a few different probes to take a global characterization of the gels. We try to with a kind of sharp probe how the particles attract to the gels, and so on. But really, it's still difficult to understand and find a good dependence, what is what. And we go, in this case, to the principal component analysis to understand really what from all these components are influenced to the hydroxyapatite production. And we was very surprised that really here it is, you can see in the plot, the red is hydroxyapatite, and the green, it is components which are more influenced to the hydroxyapatite production. It is a kind of cell viability, cell density, which understand, and the amount of the calcium. And really, what we are more surprised at the Young modulus of the hydrogel, as it was written in a huge amount of the papers, don't have such strong influence to the hydroxyapatite production, much more less than the particles. And for example, the amount of the magnesium ions, it's quite understandable that they're not influenced, because we have a balance. If we have less magnesium, we have more calcium. Calcium is nice. But Young modulus of the hydrogel, we was very surprised. In this case, we start understand step by step what influence, for example, for the hydroxyapatite production and cell growth, and understand for each pairs of component for cell growth almost always influence or size of the particles or Young modulus of the particles or amount of the calcium of the particles, and completely don't influence total Young modulus of the system and total Young modulus of the hydrogel. And if the size of the particles is so important, what influences the size of the particles mostly? And we understand, size of the particles influence if we have much more calcium in it. In the case, really the total figure, kind of total picture, it's like this. If we have more calcium in the initial system, we have bigger particles, maybe have more mass particles, more size particles, which cells grow better and provide better hydroxyapatites. In the case, in next step, we try, okay, let's take just calcium carbonate particles, simplify our system, completely remove the magnesium, yeah, and check how our gel work. If we have normal gels, which nothing grows in it, or thermal kind of annealed gel. And we do more or less the same study, but more detailed to find proof of principle. What is the reason why the cells like growth on such gels and don't like to all others? Why it's we need such strange procedure like the preheat gels to do synthesis exactly in it and grow cells, but it's not work, for example, when we just take gels, not preheat, and don't add the particles. Yeah, we do this analysis and understand, okay, really the just combination of the thermal annealing of the gels and adding the particles work well for the cells. And after this, we do a very kind of complex analysis with a lot of methods like the atomic force microscopy overlapped with confocal microscopy, together with kind of force measurement with atomic microscopy, and realize that the really cells attached just to the particles and not attached completely to the hydrogel. It doesn't matter how the strong gel is. And in this case, it is very important that the not really the size and shape and so on of the particles, but particles just provide kind of amount of the centers which cells attached. In the case, if we have enough concentration of the centers, enough surface of the particles, cell will be attached well. And what is more important that them for such kind of biocomposites, that's how strong particles attach to the gels. Because we do the control experiment, for example, for this, if we just put the particles on top, they not work. We need minerals inside that it will be really inside the gel structure. In the case, in our component analysis, the young models of the particles, if you really with the needle of the IFM with the tip of IFM pressed to the particles, this one amount should be stronger. That it's not pressed down, not float and so on. They really fixed in the wet hydrogel. In the case, all rest property like the how the strong our hydrogel, how it's dense, it's more or less doesn't matter. Depends on material property. Yeah, really, it's why I talk about this, because to summarize such application, we can provide the kind of gels which are loaded with the particles, which different functions, and it has potential much more than the just bone implants. It's if you know how to combine the gels and particles as function and the living materials, it could be wound healing, for example, the gels, or another coating, for example, for antibacterial or probacterial, or at least use as ink for 3D bioprinters, or many other kind of possible applications somewhere when we need to combine them. Yeah, and as conclusion, what I'd like to say is that it is okay. In our work, in our lab, we know how to create many different particles. We know how to create gels based on these particles. And we know how to create kind of biocarrier, bio-object based on these particles mixed together with cells. For example, and with any possible combination, like the particles inside the cells, particles near the cells, cells on top of the particles layer, cells in gel with the particles, cells on top of the gels on particles. Yeah, and here it is, we use the particles, sign the pores, we can anytime load with the possible functional molecules like alkaline phosphatase enzyme, as I show in my presentation, but it's possible to be any molecule. Okay, yeah, I think it's all. Thank you for your attention, and yeah, I will be happy to answer any question which you have. And sorry for technical problems.
Thank you very much, Bogdan. No worries, no worries. I think we got everything running, and also for the next presentation we can use this same setup. So, any questions, please go ahead.